The next amino acid in the list is alanine. I did want to mention that I'm making subtitles for these videos, so if you're having trouble understanding or following what I'm saying, these subtitles should help. I'm also putting minor corrections in for when I make a mistake, such as calling the side chain a functional group. That was a mistake from a previous video. Okay, you can see alanine here, second in the list after leucine, again sorted by abundance in protein. So roughly 9% of amino acids and proteins are alanine. And I did want to mention the reference here will tell you a little bit more about how they came up with these numbers, but they took about 5,000 proteins across all kingdoms of life, all domains of life, and of those 5,000, about 600 are eukaryotes. And again, the eukaryotes are pretty close to the total, especially just in order. And if you're watching these videos as a playlist, it might get a little repetitive, and I apologize, but I will kind of explain the same things over and over. So I'm going to draw alanine from scratch on this MolView website. So we have our alpha carbon here with amino group and carboxylic acid group. And I'm drawing it as a neutral molecule, whereas in a cell this would be a negative charge without the hydrogen, and there'd be an extra hydrogen over here with a positive charge. So this is the starting point for any amino acid, and with alanine you have a methyl functional group as the side chain. So let me just show you how they draw it when you search for it. Here's the alpha carbon, the side chain, and here's the 3D representation of it with the N terminus on the left and the C terminus on the right. It's, other than glycine, this is essentially the smallest side chain. It's a very simple just methyl group. The abbreviations for alanine, ALA is the 3-letter abbreviation, and just an A for the single letter abbreviation. Alanine is a non-essential amino acid. In order to talk about the metabolism, if you go to the glutamic acid Wikipedia page, you'll see here metabolism converting alanine to glutamate, which is glutamic acid ionized. So alanine combined with alpha-ketoglutarate can produce pyruvate and glutamate. And if that doesn't make any sense to you, just know alanine is an amino acid, glutamate is another amino acid, and alpha-ketoglutarate and pyruvate are mostly associated with the citric acid cycle for metabolism, but they're definitely common molecules that'll come up. I guess one thing to talk about, we started with leucine and then now we're on alanine, and it's kind of interesting that together they make up roughly 18% of all the proteins, 18% of the amino acids in the proteins. And so you probably are wondering why it's a decent chunk, 2 out of 20 should be 10%, but it's closer to 18%. And I think this might kind of give a little bit of a picture. Alpha helices are secondary structure of proteins. The primary structure of the protein is just the sequence of amino acids. Secondary structure starts to form into a shape, and you have alpha helices and beta sheets. This table here on the Wikipedia site for alpha helix, if you sort it by helical penalty, you can see alanine is 0 and everything else is positive above alanine. Helical penalty doesn't really mean much to me. It probably doesn't mean much to you, but it's a measure of the free energy change. So they set alanine to 0 just as a baseline, and if you were to have an amino acid, arginine or leucine, you'd have slightly larger free energy change going to that amino acid, and it states right here, higher numbers, more positive free energy changes are less favorable. So alanine is actually the most favorable amino acid in an alpha helix, with leucine being close second or third. I guess it's tied for second. So I didn't mention this, but alanine, the side chain, is a hydrophobic, non-polar, non-charged side chain. And I think it'd be helpful just to talk about that for a second. So this is a Wikipedia page, electronegativity, and there's the table for electronegativity for all the elements. And you can see carbon here about 2.55, hydrogen about 2.2, and they're relatively close to each other. So when you have the side chain here, the hydrogen and carbon are going to basically equally pull on the electrons between them, and you wouldn't necessarily find the electron closer to one or the other. There's a cloud all over the place, but there's no drastic probability of finding it closer to the one atom than the other. But if you look at oxygen versus hydrogen, you can see oxygen 3.44, hydrogen 2.2, and oxygen is going to have a stronger pull on those electrons. So compared to water, I'm going to just draw a little water molecule over here. The water molecule is going to be charged. Oxygen will be more negative. The hydrogens will be a little bit positive, slight delta charge on them. So you'll have a polar molecule over here, and all the oxygens are going to kind of stick together because they're attracted to each other because of those charge differences. Whereas when the water gets close to the methyl group, there won't be any reason to stick to it more so than just other water molecules and other charged and polar side chains from other amino acids. So you end up with a hydrophobic group here, and I think that might be somewhat useful for the alpha helice to not have too much charge on the outside. I guess maybe that's not true. Some of these are charged here. But you'll definitely, form, when you have a protein folding up, the tertiary structure is going to be how it folds. So you get kind of pockets of hydrophobic areas in the protein, and if you have a protein that's spanning a membrane, all the hydrophobic areas will want to be inside the membrane in the middle of the lipid bilayer, and the charged side chains will kind of be outside of the membrane towards the inside or outside of the cell or compartment. I think that's pretty much everything. Again, alanine, what I'm showing here is the left-handed version. There is the left-handed alanine is the one that is incorporated into proteins, even though the right-handed is showing up in different places. it's not going to be in the proteins.